in the point of view of having asymptotic stochastic chemical reactions and to be able to have asymptotic results. So, asymptotic computations. This we have seen computations. We have seen during this meeting basically how to use Gillespie algorithm to simulate a chemical reaction where you have A plus B that gives AB in a context where there was not necessarily positioning space. It was an abstract chemical reaction. So let me just come back to this. Well, this is the introduction. And then I'll state where we are going and what we would like to calculate. So basically, to characterize this events, you need two important quantity. One is called the backward chemical reaction, and the other one is the forward, and the other one is called the backward. This is called forward rate, and this is called the backward rate. Now, backward rate. So these quantities are linked to chemistry. We know that k minus 1, and this we have seen in class, this is exactly 1 or equals to 1 over the mean first passage time, or twice the mean first passage time, to escape a potential world when we understand that the process of AB that leads to the dissociation is escaping from the landscape, let's say, of a potential, let's call it VA, and this is the distance, this is the radius. You have the two molecules. One arrives and drop into the potential, which is electrostatic, and in whatever the electrostatic here is involved, it creates this effective potential. And usually this time here, this is the escape time, which is the time when the B particle is in here. It is the time it takes to escape, going up to that point, and the factor 2 is usually because there's a certain probability that it comes back, so the escape rate is really you fall into the free space. So this is the backward chemical reaction from the theory of the Kramer theory. So this is how it is calculated. Experimentally comes from all kind of biochemical measurements. So this is, and the physics is described here. The reason why it can go over the potential is because of the noise. The noise is a driving force. So this is for one, this is the backward binding rate. Now, what is the forward rate? The forward rate is the rate at which the two particles meet. And so, for example, if they are inside a domain, or if one molecule, so you have a set of collection, let's suppose site B are the site of immobile molecules, or traps, or binding sites. So those are the B molecules. And now there is some A molecule here, so this is A, and this is B. And now really what is the forward rate, if you think about one molecule, this is the rate at which a molecule will arrive, if there is only one molecule here and one binding site, this is the rate of arrival here, which is exactly the narrow escape time. So usually K1 is 1 over the narrow escape time that we have studied in the previous class. So NET is, so let's call it tau NET. This is the time, tau NET. This is the time we calculated. This is the mean first passage time. Mean first passage time. Mean first passage time to a small target. And then we have seen that we have given some asymptotic result. the week before, if you're in dimension 2, dimension 3, that we have tau 2D, tau 3D, which is omega, which depends on the geometry. Again, I'm not going to enter into the discussion of this. This is what we have seen previously. So this is in dimension 2, where omega is the volume, epsilon is the size here of the ratio of the absorbing to the total boundary. And in dimension 3, this was omega divided by 4d, and this is the diffusion coefficient here, pi d, and here this is 4d. A, where A is the size of an absorbing, the radius of an absorbing disk. So this is of this order, where A here is very small, and epsilon here is also small. And of course, when we have K binding sites, or M binding site, and S of this molecule, the rate depends on how many molecules are already bound. So the rate, the general rate, when we do, and I will come to this in a few minutes, depends, when you have an ensemble of particles, depends on the sites, total number of sites, the number of sites that are occupied, because the particles, the binding will only occur with the sites that are free. All right, so today I would like to go into the asymptotics of this, not only simply doing simulations, but how do we get, you can answer the following questions. So those are the questions we'd like to answer. For example, can we calculate at steady state? So question number 1, how to compute at steady state the different moments, that is, if we call AB the number of bound molecules, let's call it, let's suppose this is time t. So this is a stochastic number, because particles, basically we will see that in the limit where we have small targets, because escaping here from a potential when the well is deep enough, the escape is Poissonian. So the rate of escaping, so this is going to give a Poissonian rate. So this is a local process, and when this is deep enough, this is only characterized by the first eigenvalue, which is 1 over the mean first passage time, to go out. If the sites are small enough, and they don't accumulate into a big site, so they have to be, the total number of sites should be small enough. We have seen that the rate of arrival here, the probability that a particle reach one of the sites at time t is also Poissonian. So it's also proportional to an exponential lambda t, where lambda is exactly equals to 1 over the mean first passage time to mt. So under these conditions, that the number of particles, the sites form a small ensembles, this and these are both Poissonian, and from this we will be able to write, these are the assumptions. Then under these conditions, we will be able to write Markov chains, and I want to explain how you analyze the Markov chains to get information about the different questions that we're going to ask. What I wanted to say is that sometimes, if it's not Poissonian, then it's much more difficult to analyze. And let me just give an example. So we will see that this is more or less the general framework, but sometimes it is not working this way. This is when you have a polymer, like a DNA, and you look at the rate, for example, of a particle. If the particle has to bind here key sites on the DNA, a molecule can slide along and can move away from the DNA. But since it can stay confined in a boundary layer of this, that we should not, it can stay confined here, the rate at which it is coming back is not single exponential. And that creates a difficulty in the analysis of this type of problem, right?
Why is it the confinement that breaks the single exponential?
Because you stay in the boundary layer. And if you're in the boundary layer, when you expand the solution of the diffusion equation, you have not only the first eigenvalue, but you have to have also the second one. And then it becomes more tricky. I'm not going to consider this. We've tried several times, and it's not that easy. Unless the particle escapes the boundary layer. That's why this is really important here, because the idea is that the dependency on the mean first passage time is true if you start on the boundary layer. It is if you start very close, because you have the time to find it is not going to be any more single exponential because of this. It's like making short time asymptotics in diffusion. But most of the case, you are outside the boundary layer, so you can go to this limit.
So the point is, for the exponential to kick in, you sort of need it to be a long enough time that all of the, it's larger than all the inverse eigenvalues except for the first one.
That's right. That's right. That's right. This is very important, this notion. Again, there are problems where you can find it, and then it's very tricky. I don't know of any good analysis of this. All right. So let's ask the question, what is the mean number of particles of bounds at time t? And actually, what are any moments? We want to calculate any moments of these quantities, that is this to some power k and t. T can go positive, it can be infinity when you're at equilibrium. So you want to be able to have this information. So this is one type of question. And you want explicit formula, how it depends on the total number of particles of B, total number of particles of A, how it depends on the different rate, K1 and K minus 1. Now the second important question, which occur in the context of biology, it's about activation. What is the meaning of activation? Is like there is a molecular process going on, and when a certain number of sites are going to become activated, bound, you are going to see a physiological response at another scale. So for this is related to what is called the mean time to threshold. Mean time to a threshold. Mean time to threshold. Which means, how long do you have to wait in average? So let's define tau t. This is the first time in this process that we just described that the number of AB molecules, which is a random process, random variable, it's a stochastic process, the first time that this is equal to the threshold t. Condition, let's say that you started initially with AB number of molecules, of bound molecules, AB at time 0 equal to 0. You start no binding molecules, and they are all the A molecules in red. They are all here. And then each time you have bound in blue, you count it as one event, and then you are interested in, for example, suppose you have 10 binding sites, so you have 10 of them, and you're interested when is the first time you're going to have 5. So how do you calculate this? What is the first time you get this event of 5, and this is an important event, for example, in plasticity. We know that when calcium enters in microdomains such as dendritic spine, if during this realization a specific molecule called kinase 2, which is an enzyme, an oral enzyme, with specific shape, if you can activate it, which means that there's a certain number of calcium bonds to this molecule through another molecule, which is called calmodulin, then you activate processes that lead to changes in the structure, and we think, the community thinks that this is the beginning of memory, the beginning synaptic plasticity, the beginning of changes that will last for hours, although the initiation is hundreds of milliseconds. So this is how you go from a time scale of millisecond to a change that will occur, that will last for hours. And in our brain, in vivo, this is like when you slice, you do that on cultural slices, but in the brain, when we learn something, it lasts usually, it can last tens of years. So today, it's still not understood how you go from molecular events that occur in these microdomains that leads to these changes in the structure that will encode changes that should encode for learning. This is still a very active field, and the theory in this field is very elementary. It's just beginning. We're just trying to start to position, to see how we can relate this event using a classical chemistry into some a framework where we can say that going from molecular level to the cellular level is, the interpretation of this is you need to have a certain number of binding sites that are bound to the with your molecule of interest. That's why many years ago we defined this mean time to threshold, which is the first time AB equals T, and the goal is of course to calculate the mean. So you take the expectation of all realization, and what you are interested in is of course to see how this time depends on the threshold. So how it depends of T and the number of molecules, the initial number of molecules, let's say A0. This is A0 is the total number of molecules, B0 is the total number of molecules B. So A0 is the total number of molecules A, and B0, total number of molecules B. So you are interested in how it depends on T explicitly. You want to see if there is some kind of general behavior.
You're trying to go in the opposite direction of time as our distinguished visitor from Yale, who's trying to explain how we can think faster than Donald Trump by making the neurotransmitter release happen faster than the vesicle fusion in other places, right? There's some kind of, he was talking about there's a natural time scale in which these cellular processes happen, but in the neurons it's faster, in the synapses it's faster, and now you're going the other direction, which is how do you go from the natural time scale of cellular processes to learning time scale?
That's right. That's right. Because, so, over the last 40, 50 years, so now this is to answer your question, this is parentheses, here's the dynamics, this is synaptic connections. This is the synapse, neuronal synapse. So, time zero, an action potential come, you have a vesicle, the vesicle release its content. Neurotransmitters are flowing. Here you have small receptors for some specific condition. Calcium ions are flowing here. We're talking about this event is about, it starts time zero. After less than a millisecond, your receptor have been they have already left the synaptic cleft, they have already opened the receptors. The flow is almost immediate, and then here you have some specific molecules, and these molecules are sitting. The time scale of calcium, the residence time here, we actually discussed it last time. This is related to the first eigenvalue of this operator, which is the mean first passage time to escape from a spherical domain connected by a pipe. This is when you calculate and when you do experiments, is about 10 to, let's say, 50 milliseconds, 100 milliseconds maximum. After 100 milliseconds, that everything is gone, you want to ask, how many of these molecules have been seen by calcium, have been able to bind a calcium molecule, a calcium ions? So for example, after everything is done, you can say, all right, I had 5 out of a certain number of them that were bound to this calcium ion. Now, if you reach the threshold, that's it. You say now, and this is what people have observed, you can see some changes. The next time you do these experiments, instead of having a current, this is time, and this is the current, instead of having a current which was like this, when you did this, now you have a current, it is like this. And this lasts for hours when you're doing it in the slides, in the laboratory. This is permanent. Now something has changed in a permanent manner due to these initial events. But I'm saying that this field is still very immature in terms of the modeling and understanding of how this is. So we started this, and so this is exactly to answer your question. This is about, let's say, a time scale of maximum 100 milliseconds. And these events will going to stay for hours. So this is usually very hard to understand the change of scale. The time scale changes. How do you learn something like in a very short time and the thing will stay? This is the point of all of this type of analysis. So there are still many, we're still working on, for example, you can ask, and this is later on, that's why there is also something important here in these calculations, in this idea of asymptotic calculation. You can ask, how this depends on the geometry? If it depends on the geometry, we have to see exactly where the geometry enters here. Right? So this is the question that I have in mind, is how the mean time to threshold t depends on the geometry of the domain. Geometry means usually it comes from the expression of the mean first passage time that we have seen here, that accounts for that, takes into account geometry. There is a little bit, there is something else I have to add. I'm not sure we'll have time to discuss this. There is also an important feature is the relative position of the binding site between themselves. This is much harder because this is, again, you can take this into account in the mean first passage time, but then this is the mean first passage time not to one target, but to a given number of targets. Okay, so let's go back to here. We're interested in any moments, mean time to threshold, and applications. And the application of this, so the application of being able to calculate this time, as we have said here, we have neuroscience. This is the induction. I mean, again, it's a model. I'm not saying this is like this. This is the vision we have, that it can happen like this. And if it happened like this, then this is the quantification we need to say, for example, now, depending on this number, this molecule is called calmodulin kinase 2. You know, you can ask, and this is really a relevant biological question, how many of those molecules you need to guarantee that you can induce in a reliable manner plasticity? For example, if you have 3, if you have 5, if you have 10, this is why the asymptotic expression is important, to be able to answer this question in a quantitative manner. So induction of synaptic plasticity. And then a couple of years ago, we looked at some application really in, I would say, genetics or cell biology, which was with Kevin, and which was about this messenger RNA escaping from the nucleus. So what is the meaning of this? We looked at the following question. Suppose this is the nucleus of a cell. We have a messenger RNA. And we imagined that this messenger RNA could bind basically to two type of molecules. There was this binding sites, and this is the binding sites on the messenger RNA. And there were two types of binding. Let's say there would be a binding here that would prevent the messenger RNA to be degraded. And there was another type of binding. I don't have any other color. Yes, I do. And this was to destroy, to kill. So if this one was bound to this site, and if there was enough of them, then the messenger RNA was destroyed. I'm not going to enter into terminology, just to explain the concept. And so we were interested in how long it takes for this structure. So as long as you have this molecule that protects, if you have one, this site is occupied, this cannot come. However, so for example, if this molecule is bound all the time, and we are interested when this is going to escape, let's say somewhere, this is the nucleus. If you have this binding, this configuration, and if all the time they are bound to the messenger RNA, it will escape alive. Now there is a competition, because if you start when you create the messenger RNA, you have a competition between the good guys. So this is the good guy that is protecting. Good guy, and this is the bad one. So if you have a certain number of them that are bound here, that's it. It is degraded. Now, what's the probability that this will escape alive? And so this is again, this falls exactly into this ensemble, this structure of you need to ask when they can bind and unbind with a certain rate. When this guy again can bind and unbind, so we have this binding with K minus K1 and K minus 1, and we have this good guy again that can bind and unbind with K2 and K minus 2. Depending on all of these rates, what's the probability that it will escape alive, which means what's the probability to escape, or to when it reaches the boundary, with a number of bad guy, which is then the threshold T. Right, this is to show that this framework can have different type of application in cell biology, and I'm sure in other biochemistry or cell biology that can be adapted. All right, so now this is where we are going. Now let's start then to write the master equation, to analyze the master equation for these questions. And I want to ask one more thing, which is going to be the second part of the talk. So to approximate this, so let's have this in mind. This is really what drives us. Suppose at time t equals 0, you have particle that can enter your system, and there is here a place where the particle can escape. So basically, you never reach really equilibrium. You have really a transient process, and you are interested when everything is finished, really about the number of sites that were bound. And so this is now a probability, because sometimes all the particle comes, and if a threshold is not achieved, then the particle continue flowing, and we have our probability will be 0. Of course, when we average over many realization, we can then calculate, at least empirically on the computer, this, but we are interested in having this probability, being able to calculate this, again, as function of the different parameters. All right, so this is really the motivation. So it took us probably 10 years from the moment we started, started really to have some ideas of what to do. So what I'm going to summarize today, this is published, this has been first published by Zev Schuss and myself. This is Journal of Chemical Physics. This started in 2005. Then we had the second paper with my student Can Dauduk. Then this is Phys Rev E. This was in 2010. Then there was a third paper in, I think, BMC. So again, this is Dauduk and myself. This was BMC, probably biophysics, in 2012. And this is an application of this for when two cells have to separate. You need to have a process where the chromosome have to align. And here there is exactly a time to threshold process, because you want to make sure that all of them have bound to a certain molecules before it detaches. And so we could infer some condition on this rate constant based on this analysis. And then what we did is this work with, so this is Kevin Burrage, and there was other people, Ellen, Ellen and Byrne, yeah.
Anna Jones.
Yeah, and there were other people. So this is Journal of Mathematical Biology, and I think it appeared in 2015. All right, so this is going to be the bibliography for this work. Okay, so now let's start with the equations, and I think it's important to write them and to see how we're going to solve them. Yeah, it's probably a long introduction, but I think it's important to clearly understand what we're doing, why it is our motivations, and how we are going to address them, how we're going to calculate. Okay, so now what do we need? So let's start. Now this is part one. Now we are going to consider the case of a closed ensemble, a closed reservoir of, let's say, particle, and what was the name here is M and S, which is A and B. So this is M and S, so this is with the rate M plus S gives MS with the rate lambda, and K minus 1, this is for the unbinding rate. Now what is the probability? So let's suppose we start with M0 molecules at time 0, S0 molecules at time 0, and we are interested in the probability that the number of binding sites, MS, is equal to K at time t, and let's call it pk of t. And let's calculate, let's see what is the Markov chain, and what we can extract from this. So how do we derive classically the Markov chains? We just see what happened between time t and t plus delta t. The probability that we have, so now let's look at the picture. We have the binding site. We have, this is the sites that are bound to a molecule. And suppose we have k bound sites at time t. K bound sites, it means how many molecules we are left in here. So it's m, total number, minus the number of bounds, which is m minus k. And if we have S molecule here, the number of free sites, so this is bound, and this is free. So the number of free sites is equal to the total number, S0 minus K, right? This is just elementary consideration. So what happened between time t and t plus delta t? There can only be one event at a time. So, between time t and t plus delta t, to arrive to k binding site, either we started with k minus 1 binding site and the molecule arrived, or we started with k plus 1 and 1 left, one is gone, or we started with exactly k and nothing happened. So this, now we have to write it. So this is the probability that we started with exactly k at time t multiplied by the transition probability when nothing happened. Nothing happened. Or this is the probability that we started with exactly k plus 1 molecule at time t. So let's write what is the transition when we have k plus 1. It means that there was k bound molecule and one molecule is detached. So 1 out of k plus 1. So what is the rate of detaching is k plus 1. What is the rate of detaching when we will have one molecule? This is exactly k minus 1. And this occurred during time step delta t. Or we had k minus 1 molecule that were bound. And now what is the rate that 1 is going to arrive? Well, we have to see how many molecules are in this bulk. So it's m minus k minus 1, so it's minus k plus 1. So one molecule here has to choose between the sites that are free. So how many free sites we have? When we have k minus 1 molecule, so the number of free sites is S0 minus k plus 1, because it's S0 minus k minus 1, multiplied by the rate at which it happens, which is lambda, multiplied by delta t. And now the probability that nothing happened is 1 minus the probability that something happened. So let's write what is this nothing happened here. So this is exactly 1 minus, now we start with exactly k and nothing happened, so this is minus k minus 1 delta t. And when we start with exactly k, this is the same quantity here, but at position k. So minus m minus k, S0, so M0, S0 minus k, delta t. So how do we derive from this the Markov chain? We take this guy, we'll put it here, so we have t plus delta t minus t, and we have delta t in here, and so we then will divide everything by delta t. And when we do this, you end up with the classical Markov chain for this process. So let's write the equations plus the boundary. The boundary is also important because if you start for k equals 0, there is no binding site, then there was nothing, there was no molecule bound. So then we can only go in one direction. So which means that this k minus 1 doesn't exist when all the molecules are here because there is no binding site before. So we have the same equation except that this one doesn't exist. And when all, let's suppose now, let's make an assumption that there are more binding sites than molecule M. When all the binding sites are already filled, you cannot fill another one because there is none. So then this arrival, k minus 1 doesn't exist. There is no more space for one to arrive. You can only detach. Okay, so now let's write this. So we end up with a system of equation which is p dot 0 equal minus lambda m s, so m0 s0 p0 plus k minus 1 p1. Now let's assume that we have more n than s. If otherwise we'll have to have consider the equation in n, so we consider the equation in total S0. This is S0. So let me write, S, sometimes I will forget the 0, so S0 means total S. PS0, this is K minus 1, S PS, again P0 S0, plus lambda M minus S plus 1, PS minus 1, and in the middle we have exactly what we've written here, pk equals minus, so here I've forgotten lambda. So this is minus lambda m0 minus k, s0 minus k, plus k minus 1, pk, plus k plus 1, k minus 1, pk plus 1, plus k plus m minus k plus 1, s minus k plus 1, lambda pk. pk plus 1, pk minus 1. Okay, so now, of course, this is an equation that is linear, and if you multiply everything by lambda, it's also a solution, so we need a normalization condition so that there are the probabilities. So the probability to make sure that you are one in any states of this, we have to normalize it to go from 0, let's say, to s of pk of t at any moment of time equals 1. Okay, so this is, in principle, if we know how to solve this equation, we can get any information we want about the statistics about any moment of interest, right? So for example, so now let's do some remark. So this can be looked at using the Gillespie algorithm. And all the variants of this, so we can run simulations and get, again, all kind of statistical quantity from this. It doesn't mean when we run the simulations that you understand the dependency in the different parameters. Usually you have to explore the parameter space at the expenses of running your simulations in the region of interest. And second, so we can calculate not directly. No, you can calculate from this the different moments. I cannot read what I wrote. It happens sometimes. Yeah, so we are going to see now how we calculate from this. So you can calculate, you can compute the mean time to, let's say, to a certain threshold, which is what we have discussed already. Compute mean time to a given threshold. Okay, and so, of course, for the moments, for the steady state moments, so there is remark number 3, we can calculate any moments of interest. So the moments can be calculated. So if we are looking at the steady state moment, steady state is when we take t goes to infinity. Well, we can calculate, for example, the mean, which is pk infinity, from k equals 1 to s. So this is exactly the mean number of ms molecule. We can calculate the variance of ms, which is given by k squared to pk to infinity minus k to k, but minus the mean square really. And any moments of interest can be calculated if we know how to solve these equations. So let me go to the steady state. How do we solve this? What is the solution? The solution at steady state can be calculated by induction because, so this is now the steady state distribution. So how do you calculate the steady state distribution? Take dots, everything in dots in the derivative with respect to t is equals to 0. So then from here, how do you calculate by induction? If this is equal to 0, p1 becomes a function of p0. So you write p1 as a function of p0. Then we go back to p2. p2 can be extracted from here. p2 depends on p1 and p0. But since you know p1 and you know p0, because you just express p1 as a function of p0, p2 is a function of p0. And then you continue like this. And this is a routine elementary calculation that you come up with the exact formula. So, P1, what is equal to P1? P1 is a lambda M0 S0 divided by, now I need your help to make sure that I'm not doing any mistake. So P1 is equal to this, right? Okay, and now you can, I'm not going to do all this calculation, but you can calculate P2, P3 and P1, and Pk is actually equals to lambda k as divided by factorial k, k minus 1, and then you have the product, no, k minus 1 like this, I'm sorry. I have to be careful now because k minus 1 means the backward binding rate. Lambda k, yeah, pk yeah, to power k. And then we have the product of m minus I, s minus I, from 0, product from k equals or I equals 0, to k minus 1. And that's it. Now, for the no, and I'm sorry, multiply by p0, because this is, you have the p0, so this is for any k, you can calculate it up to ps0, and then you use the normalization condition, which can be written as p0 plus the sum of pk for k equals 1, let's say to s0 equals 1. So p0 is p0, whatever it is. All the pk here depends on p0, and so this leads to the equation you need to have an explicit expression of your pk as a function of the rate of arrival, which is the forward binding rate, the backward binding rate, and some combinatorics. But in principle, we have the geometry is contained in this lambda. And we have the entire expression. Now you can use this to inject into all kind of any of your moments of interest and calculate whatever you want. I have, so the problem basically here is solved because it's not that difficult. We can do everything at steady state if we're interested at steady state and the steady state regimes and the statistics. This is why we started all of this. I didn't plan to discuss this, but let me say why we started to calculate this precisely. This was when we started to work on phototransduction. Phototransduction is the process by which a single photon arrives into what is called a rod photoreceptors. So you have a rod which is, let's shaped like this, and then you have the synapse. So this is called the outer segment. This is called the inner segment. And here you have the synapse. So this is in your retina. The photons arrive like this, and they have to hit a rhodopsin molecule, which is a molecule that can detect light by changing conformation. And so the question is, what was the noise of the photoreceptor? What's the meaning of the noise? Is that usually if this is time, and if you look at the voltage or the current, you see that, if you just look, so channels here open and close all the time. And the opening and the closing of the channel is exactly due to the binding and unbinding. So you have here some channels, which are called cyclic GMP channel, whatever the name, channels. And so these channels are open, and you need 3 molecules to be bound to the admin, in average 3 per channel. Otherwise, the channels are closed. And these molecules here, this is called the cyclic GMP molecules, they are created and destroyed. So there's an additional process here that I haven't discussed. Is the fact that these molecules here are not fixed, but they can be generated and destroyed. And so if you stick an electrode here, the channels are open and closed. And if the channels opens and closes, it creates fluctuation in the current. So what you see is there is what is called dark noise. This is called the dark noise. And to be able to see a photon, you have to send here a photon, and there's a process which is called an amplification process, so that the cell is able to detect. And so you can see in the electrode this. So the first question we asked about 13 years ago was that how many of those molecules are actually bound to the channel? And what is the variant? This would be the first step in understanding this dark noise, because the variance of the number of bound receptors will give us an indication about possibly, and this is not trivial to do this relationship, but it's a starting point, between the amplitude of the noise. And this is how we started to be involved into this question about calculating the variance of the number of bound molecules at steady state. This is relevant in here to define what is the noise. And actually, there are 2 time scales involved here. There is a noise because the channel can be open and closed like this. It's called flickering. That is conditioned that the binding site, the channel, can still be in a closed configuration. And then there was an additional noise due to the process of binding and unbinding, which has to do with the geometry, the fact that molecules arrive and leaves, and in addition they can be randomly generated and randomly destroyed. So this was called, this process of arrival to the channel, this is called the dark noise. The flickering is about, so flickering is about the time scale of this is about 300 Hertz, while this time scale here, which is called due to the fluctuation of the arrival of molecules at the receptors, so let's call it this binding noise, is about 30 Hertz. This is what we extracted, we can see from measurements. So this is how we started to be involved in this. And then it took us 10 years to understand actually why, how comes? How can you see a single photon? And I think Jürgen Eingruber will discuss this in his talk. The models, all the steps of the model to determine what defines the noise of a system at the molecular level, which is important for our ability to see something, that a photon can go over the amplitude of the noise. So we have first to define what is the noise. And the bottom line, the conclusion, is that the same molecules that are involved in creating the noise, they are the same molecules that are involved in the amplification cascade. That is, if they are not there, then you don't have neither the noise nor the amplification. But if there are too much of them, then you get too much noise. So it seems, and now it's a bit provocative what I'm saying, but this is what we observe. It seems that there is an optimal number of molecules that nature over the time of evolution has reached this kind of optimality. And this, we looked at this by exploring the range of parameters. You can do a simulation when you put too much or you put not enough, and you can see that the response, I mean the ratio of the noise, of the signal to the noise, then decreases. All right, so this was, it comes really from this kind of motivation. And so now, before before going to the break, I think for the last couple of years, I ask this question. So there are nice open questions. So for example, now, suppose you're not at equilibrium, you have a flux. If you have a flux of the type like 1 plus sine omega t of particles that are coming. Let's say you have a constant J0, let's say. Now if you inject a number of particles here with, or suppose the mean number of particles m fluctuates with, let's suppose the total number of particles m fluctuates. This is total. So how a fluctuation, which is simply represented here by simple omega oscillations, are going to translate into a steady state. So you reach a sort of steady state regime where you are really oscillating. How this affect the steady state noise? The mean number of bound molecules as a function of omega. So of course, it would be interesting to compare the omega here, the time scale related to the omega, to the time scale of binding, which is really, of binding and unbinding, which is lambda and k minus 1. Okay, let's break 5 minutes, 10 minutes, and then I'll explain how you can use the same set of equations now to calculate the time to threshold by just truncating this equation with new boundary condition in the Markov chain. And the boundary condition that we're going to impose here, and by solving, so since we can solve it in here, I will show that you can also solve it in not necessarily steady state regime, because we are interested in the first time it takes to arrive to a threshold, which is a transient. Still, you can solve it and calculate the mean first passage time to threshold. And then we will look at how it depends on the threshold and the total number of molecules. Okay, let's break for about 10 minutes, the time that we take some tea or coffee or whatever. On air sign, yeah, that's it. Okay, so let's continue where we were. We have derived the Markov chain, the solution for steady state, and the time-dependent problem. Now let's discuss what is the meaning of the mean time to threshold. So now we want to compute what is the first time, the number of MS molecule. Now everything is closed. We will see, I hope, maybe today or the next time. So the next time will be about this, and probably I will start next time polymer models about looping. But so we will see if we can finish today this. So now this is the second part, which is part 2, mean time to threshold. So let's say we start with 0 binding, 1 binding, and this is K1, K1, or let's say lambda, K minus 1, and so on, 2, 3, and if we want to go up to the end, we go, let's say, to S bound, then let's say the total number, as we've seen here. But the idea now is to say, I want to stop the process. I'm interested in the time it takes to arrive to T, the threshold T, that is the number of MS molecules equals T. how do you write the time to arrive to this guy? Well, you have to modify the physical process, saying that this is lambda, but when you arrive to this state, are you allowed to go back? The answer is no, because you've reached the threshold. So you have to modify the physical process, and instead of having a rate to go back, now we are going to put here a rate which is 0. So this becomes an absorbing state. It is you start from here, you do whatever you do, and then when you arrive here, that's it, you are done. So this is what we want to calculate. Okay, so now let's write it slowly. So the probability to have reached the threshold before time t, which is really the distribution. So we're interested in this quantity because we would like to calculate tau, the mean time. I mean, let's define tau t, which is, again, let me define it again. This is the first time that ms equals t, condition that we started at 0. ms at 0 equals 0. There's nothing, no binding. And we are interested in the mean. What is the mean? The mean is exactly the integral. Okay, I will write it in the classical way. This is t. This is the first moment of the probability distribution function, which is the derivative of this, right? Which is ddt. If you differentiate, this is the probability that tau t is less than t. And I'm stepping one step, which is you have to integrate this by parts. You integrate this and you differentiate this. And so you get that this is equal to the integral from 0 to infinity, because the term that is fully integrated is going to disappear. And this is equal to the probability that tau t is larger than t. And you have to integrate dt. And so this is exactly, okay, so now we have to understand that this can be written, the probability, so the derivative of this, which is the probability that tau t equals t, this is exactly the probability that the number of molecules equals t at time t. And actually, the derivative is exactly the derivative that you have here. So this is exactly equal to the integral from 0 to infinity of 1 minus probability that tau t is less than t, which is this pt, et, t, probability of this is 1 minus the opposite. So this is pt of t, delta t. Okay, and now let's look at this. Now we are going to write down a Markov chain that goes from 0 to t, because we don't care about what happened here. So all of these equations are not going to be changed. They are the same, except that we need to write what happened for this guy at the state t. And now, instead of having a normalization condition like this, what are we going to do with the normalization? We go from k equals 0. Instead of s here, we stop at t. Of pk, t equals 1. Which means that this is going to be equal, now 1 minus pt, 1 is here, minus pt, I will put it in here, and I'm left with the sum to t minus 1. So this is the sum from 0 to t minus 1 of a quantity that I will call ak, the AK is just the integral from 0 to infinity of tk t dt. So, what have we done here? This equal to this equal to this equal to this equal to, yeah. So this is the mean first passage time that we're interested in calculating. So now how do you calculate this? You have to calculate the integral of pk of t. So let's go back to here and let's integrate everything from 0 to infinity. You have a linear system. Nothing is time dependent. If things becomes time dependent, this is much more difficult. But since everything, this is time independent, just integrate everything from 0 to infinity, and now we have to write the boundary conditions both in time, in the boundary conditions for this. All right, so what happened at time 0? At time 0, no particles, no sides are bound. What does it mean? It means that the probability at time 0 to be in state k, this is 0 except when k equals 0. So this is delta k0. This is 1. Now let's write down all the when, so the first equation is valid. This one is okay, doesn't change. This one is okay up to where? Well, if you plug in here, let's say t. T we have already here t plus 1, which is not good. If we go to t minus 1, t minus 1, t minus 1, we get t, which is already something that we want to explore. So this should be true if k equals t minus 2. So this is true, doesn't change. So we have the equation, I'm not going to write it down. pk equals, this is exactly the same here, for k between 1 and t minus 2. Now let's discuss what is pk of t minus 1 and what is equal to pk of t. All right, so let's see now what happened for t minus 1. T minus 1, we're here. So when you're in t minus 1, you can come from t minus 2, from here, and you can leave here. So this doesn't affect anything. And the only possibility when you are in this position is to go here. There is no return. So we need to write this down and to modify this, what happened at t minus 1. So t minus 1 is going to be unchanged for the term that is pk. So this is p t minus 1. So we have pt minus 1. I will write it in a second what we need here. You can come from pt minus 2, but you cannot come back from pt. So this term here, which is t, this is 0, because there is no return. All right, so now let's write what is pt minus 2. This should be the rate. So this should be the rate, pt minus 2. So this is lambda. We can write the rate. Okay, let's write it the m. So this is lambda m k equals t minus 1. So if k equals t minus 1, so this is minus t minus 1, t minus 1 plus 1. And then we are s minus t minus 1. This is t plus 2. This is root m t plus 2. All right, so this is this term here. Another term in t minus 1. This is going to give us t minus 1, k minus 1, plus the term in lambda of m minus k. So this is m minus t plus 1 multiplied by s minus t plus 1. So this is what we have here and what we have in here. Okay. Now what happened when we're here? When we arrive here, so this is like the end term, except that you cannot detach here because there is no return rate. So when we have to replace the S0, which was when everything was filled, that this term does not exist because when you are here, you cannot return. There is not the probability when nothing happened here because you are dead. So this is exactly 0, and we are left with a term which is like a flux, which is lambda m minus t m minus t plus 1 p t minus 1. This is the equations. Okay, these equations are the same. I'm not writing them. On 0 we have this one, and this is the new boundary conditions. This is what we are interested in calculating. So we're not exactly interested in calculating this PT. If we want to calculate the inverse passage time, we have to calculate the integral of them. Integral from 0 to infinity of pk of t delta t. Which means this is not that difficult. We just have to integrate this from 0 to infinity. So let's integrate everything from 0 to infinity, and then let's derive the set of equations that we need. So for this, again, I'm doing this calculation because ultimately what we're going to get from all of this is the exact dependency with respect to all the parameters. And which is exactly what we were looking for when we discussed about having for the induction of plasticity, how this can be obtained as a function of the geometry of the different parameters. All right. So let's integrate. We're integrating now. System. Integrating. The system star. Let's call it star. And let's call it modified star. This is star plus these conditions. So if we integrate the first one, what do we get? Well, this is, you integrate from 0 to infinity, so this is already the derivative of something, so this is p0 of infinity minus p0 of 0 equals minus lambda m s plus k minus 1 p1, not p1. We called everything a. A is the integral by definition. So this is a0 plus k minus 1, a1. Now what is p0 of 0? By definition, at time 0, we said that all the particles are not attached. So this is exactly 1 because they are all detached. So this is correct? Yeah. So this is exactly 1. And at infinity, the probability that you find them in state 0 is 0 because ultimately you are going to escape because wherever you start, this is an absorbing state. So this is exactly 0. And now we have the first equation, which is minus 1 equals minus lambda ms a0 plus k minus 1 a1. So we're going to derive basically a matrix equation. So now this is the same. We can integrate from 0 to infinity this. So at 0, all of them were 0 because this is not in this, there was no binding at time 0. And at infinity, this is 0 because, again, the only absorbing state is 0. So this is going to be 0. And this will give us a matrix. So let's write, I'm going to write the rate instead of writing all the time this guy. I will call this rate, this rate here, we'll call it lambda k minus 1. This is how I will call the rate, and then we will give an explicit, come back to the explicit expressions. Right, so we get 0 equals, so we have the rate, so this is minus lambda so we have minus lambda k plus k minus 1 k, ak plus lambda k minus 1 ak minus 1 plus k minus 1 k plus 1 ak plus 1. So this is just what we've written here. Now we have to integrate these two guys from 0 to infinity. So from 0 to infinity, this guy will give the same 0. So this is now lambda t minus 1 plus k minus 1 t minus 1 a t minus 1, and then we are only left with lambda t minus 2 a t minus 2, and this one does not exist because it is absorbing. And now we integrate this from 0 to infinity. At infinity, by definition, the probability to find the particle at time t is exactly 1. At time 0, this is 0 because there was no particle bound. So this is 1 minus 0. We integrate this from 0 to infinity. And this is lambda m. Lambda, let's call it lambda t minus 1. A t minus 1. So we have here a matrix that we can put in the format of a matrix. For A0, A1, AT minus 1, so we have T terms, and this is exactly what we need. The sum of this guy will give us the mean first passage time. And what can be shown, and this is just an induction formula, is that you can, so basically what is the idea? Here you can solve. Lambda T minus 1 is known, so you derive, put this in the denominator, and you have AT minus 1 equal 1 divided by this. So you know AT minus 1. If you know AT minus 1, you plug that in here, you know AT minus 2. And so you can continue from the arrival, so you go back in time, back in the process, to go to the first A0, which is going to be unknown. And in the process of doing this, you can show that the AK minus, that is a formula, which is AK minus 1 equals 1 divided by lambda k minus 1 plus k minus 1 divided by lambda k minus 1. Sorry, this is k minus 1, like the backward binding rate, multiplied by k, multiplied by AK. So, from this, you can sum all of the AK, and you end up with the, so let's Now, if we know what happened here, you use this formula, so I'm going to discuss the result now. So the result, what are the results? Suppose k minus 1 is 0, which means this is the case of irreversibility, that you can only jump from one to another. Then clearly the time tau of which is called irreversible, this is called the irreversible case. Then tau irreversible is going to be the sum from 0 to t minus 1 of ak. When k minus 1 is 0, this is 0, and you are left with the sum, which is clearly well known, of the lambda k from 0 to t minus 1. And now let's write that lambda k is what we said. This is 1 over lambda, sum of m0 minus k, s0 minus k, from 0 to t minus 1. I'm giving this example. This is well known. I mean, we did more than this in here. We are going to see what happens when k minus 1 is not 0. But now let's calculate. This will give us now the asymptotics in the different regime for this case. So what happened when basically this is the last arrival time. This is, you have to wait that the last particle arrived, and then you activate. So this is like a last the last first passage time. The last time you have to wait for the last particle to arrive to the binding site for your process to end. And so this is interesting because then this tau irreversible can be written as 1 divided by lambda m0 log of S0 divided by S0 minus t, and this is for M0 much bigger than S0 and t. T is the threshold. Obviously, you have a sum that you want to approximate here using sum of 1 over k. And we know that it is log. This is a classical result done by Euler a couple of hundred years ago. This is like log of n plus O of 1, a constant, which is the Euler constant. So you use this, and this gives us that the last arrival time basically depends on the log of the total number of particles, I mean this way, and lambda is the arrival rate. Of course, this is symmetric. If S0 is replaced by M0, you get lambda S0 plus log of M0 divided by M0 minus t, the threshold. This is for S0, much bigger than M0 and t. And M0 here, of course, the threshold is less than M0. And what is interesting, when they are equals, then it becomes t divided by lambda M0 S0. This is when M0 equals, well, M0 of the order of S0, much bigger than threshold. Okay, so now, so already we can see, which is what we're interested in. We didn't have to do all of this to calculate this, because this can be just seen when you have a Markov chain with just a forward rate. We knew that the mean first passage time to threshold was just equal to this, but this starts to give us the dependency in terms of the geometry, which is hidden in lambda, threshold, numbers of molecules. Now let's go to the irreversible case. When it is irreversible, that is when reversible case, which means k minus 1 not equal to 0. And then there is a full expression, which I'm not going to derive here, I just give it to you. Again, this can be found by induction. So tau, mean first passage time, which is the sum of the AK, is equals to 1 over lambda, the sum from j equals 0 to t minus 1 of factorial of k minus 1 divided by lambda, which is basically the equilibrium constant to power j, sum for k equals j to t minus 1, factorial k minus j, m0 minus k minus 1 m0 minus k minus 1 factorial, s0 minus k minus 1 factorial divided by m0 minus k plus j factorial, s0 minus k plus j factorial. Let me check that this is okay. J going from 0 to threshold. This is the ratio to power j. K goes from j t minus 1 factorial k minus j. Yeah. All right. So how do you find this? You use this formula, and you do by induction. You have to. So you can, for example, expand in k minus 1 divided by lambda. The lambda k minus 1 is related to lambda because this is proportional to lambda. So here you can see the expansion here if you think that this is a small parameter. All right. So now let's discuss. Let's do this expansion that I just mentioned. Suppose this parameter is small. It's almost irreversible, right? Because k minus 1 is very small. So you have almost no return, but still you have something. So if we look at an expansion, really a Taylor expansion, in the variable k minus 1 divided by lambda. So this is a Taylor expansion. So this is tau t equals tau irreversible for j equals 0, and plus the linear term, and this can be written here, k minus 1 am I missing the lambda? Ah, no, okay. K minus 1 plus the sum from k equals 1 to t minus 1, k lambda k minus 1. So lambda k here contains lambda. K minus 1 plus, for example, if we go to order 2, we have the term plus k minus 1 squared, sum for k equals 1 to t minus 1, k minus 1, lambda k minus 1, lambda k minus 1, lambda k minus 2, plus the term of order 3 in k minus 1 squared. Okay, yeah, k minus cubed. In principle, this is a parameter, but lambda is contained here, so this is really the parameter of the important parameter. Okay, so now what is interesting is to obtain from this the dependency with respect to the threshold. So now this becomes more tricky. You can see that now you can, for example, put that in a computer and see this, some expansion. But what is interesting is in some limit, we can see exactly the dependency. And this came as a surprise, the nonlinearity with respect to the threshold. I mean, obviously, this is the last passage time problem. You could see from the irreversible case that you have some kind of behavior here. So tau t So again, there are going to be different limits. Tau t equals basically tau irreversible. So let's go in the case m0 much bigger than s0 and t, which is the similar to this one. So this means much bigger than this and t. This m0 is much bigger than these two terms. Then what you get is k minus 1 divided by lambda m0 squared, and then you see t s0 minus t minus log of 1 plus t divided by s0 minus t. Plus terms that are negligible compared to this guy. If m0 and s0 you change this, you just replace here m0 by s0. So we have the symmetrical case. I mean, I'm not writing it here. When you exchange m0 by s0, you're just replacing this formula s0 by m0. But what is interesting is s0 equals m0, much bigger than t. Then this is tau irreversible plus k minus 1 divided by 2 lambda squared, t divided by m0 squared to power cube. All of this is why this is interesting, because it gives us the dependency with respect to the threshold, this non-linearity that are hidden here. And so I mean, this is published, I'll show you the graph. I mean, I'll draw the graph that is in here in this figure. So now if you plot tau, the mean first passage time, as a function of the threshold, again, this is in papers, but let me show you the graph. So basically the graph looks like this. As a function of t, the mean first passage time is a nonlinear function. T, and this is tau, and of course here we have fixed, so this is when s0 equals 15, m0 equals 10, we have a diffusion coefficient d, this is hidden in lambda, and you can see that you have something like this. It is, you have between, let's say, when you go to 10 here, compare when we go to 5, let's say this is 5, and this is 10. For 5, the mean first passage time was about 5, whatever units it is, this is in time, but when it is 10, the mean first passage time was about 25. Right, you multiply by 2, and this is multiplied by 5. So there is this nonlinearity which tells us something about the threshold property, the fact that if you want to wait that you have a certain number of molecules, and these molecules have to bind to a certain quantity of species, if the threshold to activation is a given number, you can see the effect of changing this threshold, what's going to be the effect. And of course, you can also study what is the effect of changing the number of molecules. This is the approach, which is usually not much pursued, but I think this is interesting because, again, it gives the dependency with respect to the different parameters, which usually you explore through the simulation. That if you have a formula, it's much nicer because you see the exact dependency and you compare with simulations. That's why it is important. All right, so this is the mean first passage time to threshold. Let's spend 5 more minutes. I want now to explain the next stage. The next stage is, you would say, all right, this is for what we've discussed, let's say, a system which is closed. But most of the systems in biology are not closed. They are transient. Something is coming, something happened, you want to look how many binding sites have been occupied, and then the process is finished. And then you ask, all right, what is in average the number of sites that has been occupied? What's the probability that during this event, the threshold has been reached? So this becomes then not a one-dimensional Markov chain, which we have seen, how to go from the different states. I've erased it, but it was on the board here. We had how to go from 0 binding, 1 binding, 2 bindings, but to a 2-dimensional Markov chain. And this becomes more tricky because we have to see how we move now in 2 dimensions. All right, I will now erase this and go to the second part. And this is exactly what we have used with Kevin to study the probability of this messenger RNA to reach the boundary without being destroyed, because we had these two, one was protecting, one was destroying. And so then we wanted to make sure that you could calculate this, and there is many possibility to escape for these guys. Suppose it can escape with 0 molecule bound. It can escape if the threshold to killed is, let's say, 5. If you can escape with 4. You can escape with 3, 2, 1 of these bad guys that are bound. So you can escape in many different stages. So this is why we need to look in this, I will show you the second Markov chains. We have to classify and to look what are the region of interest, what we want to calculate. Okay, so now let's go to part number 2. So let's work 5-10 minutes, and I think it's going to be enough for today. We'll continue next time, finishing this, and we will be able to start a polymer model next week, applied to double-strand DNA break and Ross polymer and looping. So for today now, I want to discuss time to threshold, or let's call it so Markov chains with killing. I'll explain in a second what I mean by this. This is really two-dimensional Markov chain with killing. All right, so suppose this is a picture we have in mind. You have the binding site. Time 0, let's suppose the particle enters, or they're already, you start time 0 and they are all there. And then there is a small hole. A small hole, if you remember, the time to escape through a small hole is Poissonian. So this small hole means a particle disappears. If it disappears, it's equivalent to say that the particle has been killed. I don't care where it has been killed, but it disappeared from the system. So now, we are interested, so let's suppose we call mu the Poissonian killing rate. Poissonian killing rate. So, what are we interested in here? We would like to understand, as a function of time, what is the probability that the number of bound molecules, this is number of bound, equals k, and the number of molecules in the system is m. So m is the total number of m molecules. Right, so this is the probability that ms, number of bound at time t, equals what? Equals k, and how many molecules we want to have? The number of molecules, which I call W, equals m. Condition that at time 0, ms is 0 and W equals m0. We introduce at time 0 all the molecules equals 0 and this. And the total number W at time t, this is the number of molecules that are free plus the number of molecules that are bound. This measures the total numbers, m of t and ms of t. All right, so this is a description of a system where we have, let's say, the blue particles. Some can be bound. There were more at the beginning that escaped. Some are already escaped at time t. And so we need to look at these two quantities. And what we are interested here, the question of interest, is what is the probability that at time 0 you enter your particle? What is the probability that you have T binding side? What is the probability? And when you look at the end of your process, so let's call it the infinity. This is the end when there are no more molecules. Of course, it's not infinity, it's a certain time. What is the probability that after your process is done, you have reached a threshold? Now it's a defective process because probably, or sometimes it can happen that if your threshold is 5, you got only 2 bound and that's it. So what's the probability that the number of molecules equals T? And then you can ask also, when it happened, when you have this T, how long it takes. That is, you can calculate the conditional time. The time not conditioned is infinite because sometimes it doesn't happen. So you can calculate this, and you can calculate what is called the conditional time to threshold.
In particular, capital T is not a time in your first equation.
What? I'm sorry?
Capital T is not a
No,
time.
this is a threshold. This is threshold. For example, 5. 5 out of 10 or whatever, total number, let's call it S0. This is a threshold. So now we want to calculate what is the time the threshold T is achieved. Condition that the time to reach this threshold is less than the time of killing, than the time they are all, so let's say that condition that it is less than the killing time or less than infinity. I don't know how to write it correctly, but this is the idea. You want to calculate this conditional mean first passage time. Condition that this happens. So when you run simulations, you look, each time you reach the threshold, you average, so you run one simulation. You wait how long it takes to have your threshold achieved. When it happens, when it doesn't happen, you discount this this realization, and this is how you do it on the computer. And now I want to calculate analytically. Can we calculate this quantity? I mean the two quantities. So now for calculating this, as we have seen before, we need to write down the Markov chains. Now it's not a one, but a two-dimensional Markov chains. And now let's discuss what is the picture, the structure of these two-dimensional Markov chains, and where do we locate in this two-dimensional matrix, where do we locate this probability of having reached the threshold? Okay, so now what is this matrix? So maybe we need a big word. So now we're talking, so of course if there is an additional parameter that varies, you have to study a three-dimensional Markov chains. And you'll see already with two, it's you have to look at precisely how do you go from, let's say, time 0 where you have nothing bound to the different possibility to be bound to reach the threshold T. So let's write this. So what happens? So let's write down now the two-dimensional Markov chain. So the two-dimensional Markov chains, so for this we need to understand what happened during time t and t plus delta t. Suppose you have k bounds molecule, k, and m particles. So what can happen? So first of all, how do you come to this state? Before, you could be with k minus 1 bound, and there was m particle. So k minus 1 bound and m particle means now you arrive to k. What does it mean? It means you bind now. Yeah, I'm sorry. Let's write it. Let's write it k minus 1. Let's write it this way: k minus 1 and m. So how do you go to here? You have to bind. So now we know the rate. This is the binding rate. So this is lambda s minus k plus 1 m minus k plus 1. This is the rate. Again, we have discussed this. I'm freezing now the m, the total number of particles. I just look at the events of binding or unbinding. So this we have calculated previously. Now, what can happen also is in here, you go to k plus 1, and you freeze the number of particles. And this is the rate, k minus 1. And now we have, so this is 1 bound, and now you had k particles, k bound, and now you need k plus 1 bound. So what happened for this? You get additional binding, right? So this is lambda s minus k, m minus k. Now what happened? You had k particle and m plus 1. So how do you go from here to here? So there was m plus 1 particle, and now you have m, which means that you have activated your Poissonian rate, you have removed 1 out of m plus 1. Is it correct? So this is mu multiplied by m plus 1. And then finally, you have, and I think that's it, no? Because k, no, if you have k m minus 1, what does it mean? That is, yeah, okay, now k m minus 1, which is the rate mu m. That is, there was m particle and 1 is living. Are we correct? I think so. Mu. No, I'm sorry. I made some mistake. I'll go back 1 second. M, if I have K bound, the number of molecules that are free, because you can only kill those who are free. So if they are bound, the number that are free is M total minus K. So this is here minus K. And here this should be M minus K plus 1. Okay, so this is killing. And why don't we have here, if you're bound, okay. And here I think I am in the irreversible case. That is when you're bound, you do not get, that's right. Here when you bind, I assume K minus 1 of what we have discussed here is 0. K minus 1 is 0. It's very hard if you have K minus, we didn't know how to analyze with K minus 1. All right, so that's why here you don't just go back. We assume here K minus 1, which is the backward binding rate is 0. All right, so now let's position the matrix, and then we'll continue next week to, I want to design here to show the matrix and then to position what we are interested in. So let's start. 0, number of bound at time 0 is 0. What is the total number of particles? M. Now you have to have, double check everything, but should not make any mistakes. So we agree, time 0, total number of bound, 0, total number of particles, M. Now what can happen in one direction? You can have one bound and no killing. You can have two bounds and no killing. You move on until you reach T and M. And then you are finished, because this is the state where you are interested in, right? You have T bounds, and there are more M than T. T is lower than M. So we have now finished. If you arrive here, this is a state that we're interested in. You reach the threshold. And then now we have to continue and put on the lines what happened after you killed one, you killed two particles. So now we have 0 bound and 1 particle was killed. And now we can continue like this, n minus 1, and then we arrive to here, which is T, and then we have n minus m, and T, m minus 1. We have 1 particle was destroyed, and we reach the threshold. And then we continue up to where? We continue.
You can have down arrows between any of those processes on the first 2 lines, right?
I'm sorry?
You can have down arrows between any
Okay, I'm sorry, I have, yes, sorry. Yes, this is very important, because now the arrows that you just mentioned, this is exactly what we have here. Yes, absolutely. So let's do the number. So we have 0, m minus 2. I'm sorry, I started to be tired. M minus 2, and now we have 1 molecule is bound, and m minus 2. M minus 2. And again, as Peter said, we have now arrows from this state and from this state. So we have now the arrows that goes in these directions. That is, they are irreversible. When the particle is dead, it cannot come back. And when you are bound, you cannot come back. So this is now an interesting diagram. Interesting in the sense that we can do something and trying now to find when and how we are going to get to this interesting stage. So now let's say this is 0, and now we need to go to m minus t. And then again, this is 1, m minus t. And then we are going to arrive here to t bound m minus t.
It's a finite difference scheme in 2 dimensions with 1-dimensional chains on the boundary.
Yeah, that's right.
Yeah.
That's right.
Yeah.
So now, am I correct? Yeah. This is exactly this, just numerical scheme. And what are we interested in? We're interested in calculating what's the probability that you end up here compared to when we arrive then. If you're not in here, you are not your probability is not. You're not interested in this. This is going to give you exactly the probability, all the cases, for the probability to have reached the threshold during your process. Now start the combinatorics. How do you arrive to one of those stages? So we say this is a starting point. To arrive to this guy, there is only one case, one possibility. Just go straight. That's it. Because if you do one, that's it. You cannot come back. So this is not very hard to calculate. the probability of being this. This is the probability of transition from one of these guys. Now if you start with this, now we start to see what happened, where the combinatorics is coming from. Now how you can go to this guy, you can go first this way, and then you go down. Or you can do any steps. You have to sum over all these possibilities. You do one jump, and then you move this way. And then, of course, with number 3, it's going to be the same. We have to an additional layer of complexity. You can go from here, do one jump somewhere, and jump somewhere. So then we have to sum over all these possibilities. All right? So next week, I think now I'm exhausted. Next week, I will write down the sum. Now we understand what we need to calculate, which is not very hard. Once we have done all of this, we need to calculate as a function of we'll see now the transition, the rate transition, we just calculated them. We can calculate the probability, the transition probability, for example, the probability to make a transition from here to here is going to be the rate here divided by the sum of the two rates. And so next week, I'm going to finish by this. We'll see how do you calculate this transition probability. How do you express this in terms of all this transition probability? And this will give us a formula for the probability of the mean first time to threshold. And then we will calculate the time, and the time is going to be the time to arrive in one of those states. And then we'll have to sum over all the possible paths. And so that's the combinatory that is behind. It's not very hard once you understand how things have to be calculated. Okay, so this is one example, and then what I'm not going to discuss what is in the paper. Suppose you have two types of threshold, T1 and T2, because you can bind, suppose you can split your binding sites into two types of binding sites, let's say red, and so you have one type of binding sites, this is the binding site in purple, and now we have another class of binding sites. And now you can ask, suppose you have a certain number of them, what is the probability that you reach the threshold for the first class before the threshold for the second class? So this I'm not going to enter. This is more or less the same, and now you have to split in between two type of, basically here, you have to split. And then you can ask, what is the probability you reach this before you reach this? So again, this is motivated by some biological applications. So you can have many variants of all of this. What I never worked on, and I wanted to do that at some point, and we don't have results, and I think maybe this is possibly a PhD, it's to look at what happened when the number of molecules here can vary in time. That is, you have some, let's say, fluctuations or changes in the number of molecules, total number of molecules. Let's say, for example, with the sinusoidal waves, sinusoidal functions. Okay, so I'll stop here. Next week, we will write the matrix. I mean, we have written the matrix, I will write it. And I'll finish and write the formula. And I think that's it. I think this is the basic ideas of how we calculate asymptotic expressions using Markov chains, using boundary conditions in Markov chains. Next week, I wanted to discuss actually the paper with Kevin, how we did it for this messenger RNA. We could calculate some up to a certain point because this was more involved. And at some point, we used computer simulations, but so we could still predict the behavior, which was interesting, how it depends on this bad guy versus the good guys. All right, let's stop here.